My name is Faiz Chauhan, and my thesis will talk about developing a method to analyze proteins and metabolites in human saliva. Saliva in humans is vital for protection against pathogenic microbiota such as bacteria, viruses, and fungi in the oral cavity. It plays a role in the maintenance of oral tissue health and protection of the teeth, which helps prevent decay. Finally, saliva facilitates oral functions such as mastication, starch digestion, speaking, and tasting. Due to its easy access, saliva is one of the easiest and painless biofluids to sample. Some sampling methods include the suction method, which makes use of an aspirator device, the absorbent method, which works by collecting saliva via an absorbent material, and finally, the spitting method, which quite frankly requires the subject to spit in a collection tube. The latter method was used in this thesis. Analyzing saliva allows for the screening of oral and systemic diseases, such as myocardial infarction and pancreatic cancer, just to name a few. This can be done by looking for telling biomarkers which happen to be present in saliva. Given the ease of access, it also makes it a great fluid to sample in clinical studies. Whole saliva is made up of 99% water. Other components include proteins, which consist of immunoglobulins, signaling molecules, receptors, enzymes such as amylase, and mucins known as glycoproteins, which give saliva its viscous nature. Metabolites, which function as intermediates of cellular pathways, regulators of cellular function, as neurotransmitters, and building blocks for proteins, are also present in saliva. Oral microbiota and food debris constitutes the remaining part of saliva. Although this thesis focuses on the concentration and isolation of proteins and metabolites, this work would have not been possible without the LC-MS. The LC-MS essentially allows for the separation and detection of compounds such as metabolites, lipids, and proteins, which helps identify and quantify compounds of interest. To ensure we got the best quality results, we ran the samples twice, once for the proteins using a C18 column and then a second time using the HeLa column for the small polar metabolite. Given that there is no current universal method around the extraction of proteins and metabolites from saliva in current literature, this thesis presentation will hope to provide which method of protein and metabolite extraction provides the best results. Prior to discussing the layout of the experiment, let's discuss the variables used in a little detail. Whole saliva, when collected via the spitting method and then allowed to settle, forms a two-layer solution. We had decided to give the upward portion a name and will refer to it throughout the rest of the presentation as pseudosupernatant. We called it this because although it's a liquid lying over a solid, this was not formed due to centrifugation. The image on the right shows a centrifuge sample, and therefore the top liquid portion was called supernatant. Samples were taken from each type of supernatant and used in one of the two, purification and concentration methods, to identify and quantify proteins and metabolites. The pellet seen in both images was not used in our samples because it contained mostly cellular debris and bacteria, which was not of our interest. One method we used was the bladder extraction method, which is derived from the common Folch method used to separate lipids. To oversimplify, first whole saliva is alka into Eppendorf tubes, then a combination of methanol, chloroform, and milky water are added, and finally the samples are centrifuged. This process results in the formation of three distinct layers. The top layer, called the aqueous layer, contains hydrophilic metabolites. This will be used for our metabolite data. The middle layer, called the protein disc, contains precipitated proteins. This will be used for our proteomic data. The final bottom layer is called the organic layer, which contains lipids. We will not be looking at this. The other method uses a special filter called an amicon, which works by separating protein content from the rest of the saliva by the use of a centrifuge. After performing a literature search and consulting with other researchers in the field, we determined that the 10 kilodalton molecular weight cutoff for the amicon filters was best based on the typical size of most salivary proteins. To obtain a sufficient amount of protein sample so that it can be read by the LC-MS, we started with 4 milliliters of saliva and then used 2 rounds of amicons to allow us to get to the necessary protein concentration for our proteomic data. A blind error step was done on the filtrate derived from step 1 to give us our metabolomic data. Finally, the isolated metabolites are then prepared to be read on the LC-MS. For metabolites, the samples are dried down and the standards are vortexed in, and this is transferred into HPLC vials so that they can be put on the instrument. Proteomic data is slightly different, where the samples are first reduced, then alkylated, and digested finally using formic acid. Between each of these steps is an incubation period which allows the respective reactions to occur. Then the samples are packaged into HPLC vials, after which they are loaded onto the LC-MS. Looking at our general experimental design, we started off with whole human saliva and divided this into two groups, pseudosupernatant and supernatant. These two groups were further divided into two different extraction methods, Amicon and Blydyre. This makes 4 groups, each with 5 replicates. We followed this method for both the proteomics and the metabolomic data, so we had 4 groups for the proteomic data and 4 groups for the metabolomic data. Also, for simplicity, the pseudosupernatant group combined with the Blydyre group will be referred to as PB. The pseudosupernatant combined with the Amicon filters will be referred to as PA, pseudosupernatant combined with Blydyre will be referred to as SB, and supernatant combined with Amicon filters will be referred to as SA throughout the rest of the presentation. Looking at our results, we can start off with the proteins. I initially performed a one-way ANOVA to see if there was a difference between the 4 groups, and indeed there was. When the data was uploaded onto Metaboanalyst, it was found that out of 1,026 proteins, 222 of them were significantly different from the others, which indicated that the method of extraction had an effect on the concentration of those proteins. Viewing the samples on a PCA plot, we can see that there is a strong correlation between the Amicon coupled with Blydyre and protein digestion groups and between the Blydyre only groups. However, the groups slightly intersect at the bottom of the plot, thereby showing that the effect of being derived from the supernatant or pseudosupernatant is quite minimal. To analyze this closely, a chart was constructed to compare the amounts of protein each method yielded. T-test analyses were then performed between the pseudosupernatant and supernatant groups, and these two t-tests were found to be insignificant. However, when analyses were done between the Blydyre component compared to the Amicons within the pseudosupernatant and the supernatant, it was found that the results were statistically significant. This is also evident in the protein count figure, where you can see that there's a lot more proteins that were found in the Amicon samples. These are the top 9 most significant proteins derived from Metaboanalyst, and you can see a clear trend where Amicon samples, both pseudosupernatant and supernatant, outperformed the Blydyre method by a large margin. Although statistical analyses say that there isn't a difference between the Amicon groups, by this I mean the difference between pseudosupernatant and the supernatant groups, PA and SA respectively, you can see that there's a consistent trend throughout all of the significant proteins whereby the pseudosupernatant outperformed the supernatant samples within the Amicon. So it's important to note that there was a visible difference, however, it wasn't statistically significant. Moving on to the metabolite results, a quick one-way ANOVA showed that there was no statistically significant differences between the 4 groups PB, SB, PA, and SA. Given that it was insignificant, there is no ANOVA graph to show. A quick PCA plot further showed that all the methods yielded similar results as noted by the coupling of the different groups seen at the center. A total of 20 metabolites were found. Here they are graphed in increasing peak area and in series to see any trends. One important thing to know is that although it's not statistically significant, as you get into the greater peak areas, hence the greater detection of these amino acids, a pattern can be seen where there's an increased prevalence of the pseudosupernatant group combined with the Blydyre method, also referred to as the PB method. You can also see the same trend in the results obtained from the Metaboanalyst results, where PB is outperforming all the other methods for the most part, but keep in mind, this is not statistically significant. We were able to find 10 neurotransmitters in our data, 8 of which had good quality peaks and therefore could be used for statistical analyses as shown here. The other 2 did not have consistent peaks, however, they were detected. You can see a general trend whereby the pseudosupernatant combined with the Blydyre method outperformed all the other methods, as seen in most cases, as in serotonin, carnosine, GABA, glutamic acid, and phenylalanine. However, there are also some outliers like alanine, taurine, and beta-alanine. Although such trends were seen, please keep in mind that this was not statistically significant. Finally, to gauge the amount of pathways detected when the proteomic and metabolomic data was combined, we uploaded our data onto Joint Pathway Analysis option on Metaboanalyst. Here you can see the pathway impact chart whereby the most significant and impactful pathways were plotted on the top right. Looking here at beta-alanine metabolism and amino acid tRNA synthesis, we can see that some of the proteins and metabolites in our data were found to be in these pathways, as noted by the yellow bubbles. Some other common pathways that were found were folate biosynthesis, biotin metabolism, vitamin B6, and the others listed in this chart. Although it's important to see some general pathways, it's even more exciting to see oral cavity-specific pathways, as it functions as positive control. Starch metabolism, which is done by the enzyme amylase, and sucrose metabolism was found to be the most significant and impactful pathway. This pathway usually occurs in the mouth. If you recall, sugar breakdown occurs in the oral cavity, whereas protein and lipid digestion occurs in the stomach. Another telling pathway was the mucin synthesis pathway. There's a large abundance of mucins in saliva, and they play many important roles, hydration being one of them. Seeing these pathways as one of the significant data points is interesting, but nevertheless expected. In summary, for the proteomics, the total number of proteins found were 1,026. The Amicon filters were found to be working better than the Bladire method, and the type of supernatant seemed to have no effect. For the metabolomics, the total number of metabolites found were 21, and there was no statistically significant differences between the four groups. In conclusion, the results of this project can be used to create a better method so that it may yield higher quality results. The future potential of saliva is great given that it's easily and painlessly reproducible, therefore giving it great clinical prospect. Finally, potential research limitations include the slightly small sample size and the difficulty that comes with working on larger volumes of samples. Thanks for listening. I'd like to especially thank my supervisor, Dr. Danina Simmons, for guiding me throughout this project and helping me through every hurdle. Any questions?